Good morning, everyone. My name is Momin Abdallah, and I'm a senior research scientist at Norgen Biotek. And today's webinar is entitled Recent Advances in the Preservation and Purification of Plasma Cell-Free DNA and Cell-Free RNA. In today's webinar, we're going to be talking about a brief introduction regarding the circulating nucleic acids, its origin, different forms, important findings, and its clinical applications. Also, we will be talking about Norgen's cell-free DNA, cell-free RNA preservative tubes, its specifications compared to other collection tubes currently used, Norgen's evaluation with featuring a key publication comparing 4 different blood collection tubes, including Norgen tubes. I'm going to be talking as well regarding the Norgen cell-free DNA and cell-free RNA purification technology compared to other purification technologies currently used. Different cell-free nucleic acid quantification methods, which is very important. Also, 2 key publications comparing Norgen's technology against current technologies for the purification of cell-free DNA and cell-free RNA. At the end, we're going to have a Q&A session where I can further clarify any point or answer any of your questions. So please have a pen and paper ready to record any comments or questions. I hope that you will enjoy today's webinar, and again, I apologize for the delay. So before we start, we need to understand the origin of the cell-free circulating nucleic acids and how they look in the circulation. So as you know, in the circulation, due to the apoptotic cell death or necrotic cell death, cell-free DNA and cell-free RNA will be released from these dead cells into the circulation. They're going to be either in freely circulating, bound to proteins, or contained in exosomes or vesicles. Circulating DNA in particular will be coming from normal cells. They're going to be hypomethylated, whereas the circulating tumor DNA that are coming from necrotic cell death, especially if it was coming from a tumor dead cell, it will be hypermethylated for the epigenetic changes that is happening during the progression or the development of cancer. Cell-free DNA, normally found in the circulation, they are in the form of nucleosomes, where the double-stranded DNA is wrapped around a protein complex of histones. These nucleosomes are 146 base pairs, approximately between 140 and 180, depending on the origin and where are they coming from. Each nucleosome is linked to another with a linker DNA, which is a naked DNA not bound to proteins. Therefore, when the DNA or when the cell died due to apoptosis or necrosis and comes out of the cell, the DNA itself is going to be subjected to the DNase activities that are found in the circulation. So the DNases will digest the DNA, add the linker DNA, leaving these new nucleosomes circulating in the circulation. And this is very important to know what we're going to be expecting to find in plasma, and then when we isolate the DNA, what we're going to be expecting when we analyze the purified DNA on bioanalyzer trays, for example, when we do our PCRs, all of these things. So we need to know what we expect. So since the 1940s, researchers have been working on understanding the structural characteristics of the cell-free DNA. Also, they are heavily investigating its clinical applications, especially in the oncology field. This figure provides a timeline for the main discoveries concerning circulating DNA prior to the concept of being called liquid biopsy in oncology, as well as those concerning the structural and functional aspects of circulating DNA. One of the main findings, 1977, when they found that the concentration of the circulating DNA from cancer patients is greater than that from healthy individuals. And that point, it opened the potential of analyzing the circulating DNA for biomedical applications in oncology. So publications frequency for the past 50 years showed that an increased interest by the community concerning circulating DNA and its promising clinical applications in the field of oncology, as I mentioned, autoimmune diseases, inflammatory diseases, systemic disorders, trauma, sepsis, or myocardial infarction. So the molecular analysis that are enabled by the isolation of circulating tumor cells, CTCs, and circulating tumor DNA from blood specimens may be applied to guide as well different treatment strategies and different events in the initial diagnosis and treatment of patients with cancer. That's why, in order to do this, most of us researchers and scientists are in need of a very good and robust workflow that can help them to collect, preserve, and purify these circulating nucleic acids in their highest quality and quantity. As we may know that quality and quantity are very important for all the downstream applications that we are doing in our labs. And also we have precious blood samples, so we need to have a very good and robust workflow to deal with our precious samples. So in order to do this, and as our vision at Norgen, we developed a new product called cell-free DNA, cell-free RNA preservative tubes. This cell-free DNA and cell-free RNA preservative tubes has many advantages and features. First of all, it preserves both cell-free DNA and cell-free RNA at the same time. No need to have one for DNA, another one for RNA for 30 days at room temperature, which is a very good time to preserve during storage, during shipping, especially if you are collecting your blood samples overseas. It takes time sometimes to collect the samples and then ship it. So we took this into consideration. Also, it preserves the circulating tumor cells for 14 days at room temperature. The good thing about this preservative is that it doesn't have any formaldehyde, so it's a formaldehyde-free preservative. So we were trying to avoid any cross-linkage of the DNA, which showed some issues with some of the downstream applications. Also, we developed this based on the chemistry to have a maximum plasma recovery. Our plasma recovery from this tube ranges from 6 to 7 ml of plasma. We're going to go through the rest of the features while I'm going through the in-house validation and testings. So, as you know, there are other tubes on the market like Streck DNA and RNA tubes, QIAGEN pre-analytics, and Roche. As you can see, the length of the preservation, as I mentioned, Norgen is the only preservative that can preserve both DNA and RNA at the same time for 30 days, unlike others, 7 days for RNA, for example, 14 days for cell-free DNA. Also, one of the most important features is the recovery of the plasma volume, which we recover approximately 6 to 7 ml without losing this volume during shipping, and I'll discuss this in details later. And as you know, if you want to have the highest quality, then you will directly think of isolating your DNA from a large sample volume. And as you see, the other tubes recovers 3 or 4 ml, and we will see what's going to happen to these volumes after shipping. So one of the important features of Norgen's preservative tubes, based on the chemistry that has been employed in these tubes, that it doesn't lose any plasma volume after shipping. In this study, we collected blood on 3 different tubes, EDTA tubes, another tube from another vendor, and Norgen tube. We kept some here at Norgen, and its replica were shipped and received back to Norgen after 72 hours. And then it stayed for 7 days after being received. And then we spun down the plasma or spun down the tubes to recover the plasma. So what you see in front of you is the tubes before shipping or without shipping. Stayed at room temperature at Norgen for 7 days. The same tubes or its replica were shipped, and when it came back, we spun it down. As you can see, a Norgen tube before and after shipping, it didn't lose any of its volume, so no plasma loss. Whereas EDTA tube, for example, or the other tube, lost at least half of the preservative. So if you are trying to or planning to isolate your DNA from your samples from 4 ml, suddenly you will see 1.5 or 2 ml. So we guarantee by our chemistry that it's not gonna, we are not gonna lose volumes. The reason why we are not losing any volumes, because our chemistry in this for this preservative depends on the instant pelleting of the red blood cells and white blood cells once the blood is in complete contact or mixed perfectly with the preservative. So once you mix the blood with the preservative, the composition or the components of the preservative will pull down the red blood cells and white blood cells without any centrifugation, as if it's pelleting it. And so the blood cells are not going to be moving around or floating around in your tube and the blood tube during shipping. So it's not going to take away any plasma volume. That's why we are not losing any volumes. So since it's not moving, the red blood cells and white blood cells are not moving in the blood. So the hemolysis, the risk for hemolysis is very minimal. Unlike other tubes, the red blood cells and white blood cells are floating around, hitting each other. So they may burst and they may cause hemolysis and release of the cellular DNA. And this is very important because you don't need your plasma to be contaminated with cellular genomic DNA. Also, you don't need your plasma to have lots of the hemoglobin that has the heme group that may cross-contaminate your sample and affect your downstream application, because sensitive downstream applications may be inhibited by the high amount of iron. So this is a representation for blood samples that has been shipped and came back and stayed for 30 days at room temperature. And we measured the hemolysis levels based on measuring the hemoglobin levels in the plasma at 414 nanometer. You can see that the hemoglobin levels in Norgen tubes, or the plasma recovered from Norgen tubes, remains fairly constant, whereas other tubes like EDTA started to showing after actually 1 to 2 days, increasingly very high, which means that most of the cells were degraded and the hemoglobin was released. For the other competitors, it started from 0 to 7 days. It was constant and then started increasing gradually after the 7 days. That's our in-house validation. So from these last 2 slides, the chemistry is working perfect. It's not preventing hemolysis. we are keeping the same plasma volume. So now we need to know if it's releasing any genomic DNA from white blood cells, for example. So we did, as you remember, that's why I put this slide. This is the size of the DNA that is mainly found in plasma is around 140 to 180 base pairs. So this is the main cell-free DNA in plasma within this size. Since plasma shouldn't have any cells, so we shouldn't be expecting high molecular weight genomic DNA, like the DNA that we see when we isolate DNA from cells or tissues. So it's going to be a small band at the 180 or 140 base pairs. So when we shipped it and received it back, and we did the isolate the DNA, you can see that Norgen only is showing the cell-free DNA peak around the 140 to 180 base pairs without any genomic DNA contamination. So this is flat line, means that there is no genomic DNA contamination or large molecular weight DNA. Whereas other tubes like EDTA and one of the other tubes on the market, it showed very high genomic DNA, very high cell-free DNA peak, which is due to the degradation, and also showing lots of high molecular weight genomic DNA. We have to zoom in to show this DNA or the small peak of the cell-free DNA. Otherwise, you won't see it because it was very low in concentration. Whereas because of the other tubes showed high genomic DNA or high molecular weight genomic DNA, we have to zoom in. So this is considered contamination. This is considered genomic DNA release in plasma. This is considered inefficient or as we call it, like contaminated plasma genomic DNA. It hides actually the cell circulating tumor DNA that is going to be present in very low amounts, or going to be hiding our cell-free DNA. So if you are looking for mutation or try to do a SNP analysis or anything that is very rare, it's going to be very hard to fish it out because now it's going to be masked by the large genomic DNA or large molecular weight genomic DNA. So this is very important for any of us when we are trying to do biomarker discovery or trying to do any sensitive or trying applications or trying to find any low abundant changes. So now, since we talked about the main features, this is also a very important feature, where we preserve the cell-free DNA, the small peak, as you saw on the previous slide, for 30 days at room temperature. How we assess this, the main reason we are at our main target was to amplify two PCR targets. One is a short fragment ALU 115 base pairs, which are representing the cell-free DNA, and another target, which is the long ALU 247 base pairs. This is representing the high genomic DNA. As you can see, Norgen's amplification or the CT values remained fairly constant within 30 days. EDTA tubes, the CT value started to decrease, which means that there is lots of genomic DNA is there. One of the competitors, it remained after 7 days and 14 days, and then the CT value started to decline, which means there is genomic DNA contamination. The same thing with the long fragment, 247 base pairs. The Norgen amplification for this fragment remained fairly constant for 30 days, which means there is stable genomic DNA, there is no genomic DNA release and no hemolysis, whereas the EDT tubes also the genomic DNA started to increase, therefore the CT value started to decrease. The same thing with the one of the competitors. You can see that the CT value started at like almost 18 and then started to decline by time till 14 days, and then there is sudden increase in the genomic DNA amount, which means lots of hemolysis. As for the cell-free RNA, because our tubes, as I mentioned before, it preserves both cell-free and RNA and cell-free DNA in the same tube, we assessed this by the amplification of 3 targets, a housekeeping gene, which is an 18S RNA transcript, an HPRT messenger RNA transcript, and a MER21, microRNA 21. Since the competitor only preserve RNA for 7 days or 14 days, so we tested the preservation of the competitor for 14 days, but because ours goes to 30 days, so we assessed our also preservation for 30 days. You can see for the 3 targets that the CT values remain constant for Norgen's amplification, whereas the CT values started to increase, which means the RNA that we started with got degraded and there is no more RNA, or we're losing RNA with the competitor. The same thing, by the way, for the EDT, and by the way, RNA is cell-free RNA is stable in plasma at room temperature in EDTA tubes for 1 to 2 hours max, and then it's going to be gone. It's going to be degraded unless it's very tiny, small, which will escape the RNases and the nucleases. So a few days ago, like I think 10 days ago, there was a very good publication from German Hospital Institute in Germany. They evaluated the storage of circulating nucleic acids in plasma. They evaluated 4 tubes, Norgen's tube, PacGene, from QIAGEN, Streck, and Roche. And they evaluated both the DNA preservation and the RNA preservation. And you can see for the DNA, they said that Norgen's tubes recovered the highest quantity of the DNA out of the 4 tubes, significantly higher. You can see here. Streck recovered the lowest DNA because our tubes, our tubes go to 30 days, but others goes to 4 or 7 days or 14 days. So they did their study for only 7 days. So in terms of the size as well, they found that all tubes had similar peak sizes for cell-free DNA. The big sizes was ranging from 168 to 180 base pairs, except for Norgen. And Norgen tubes was the only exception. The big size was 146 base pairs. And if you remember from the early slides, the nucleosome is of 140 base pairs or 145 base pairs. So they explained this by the fact that the length of the DNA wrapped around one of the nucleosomes, which was the 146 base pair, and this might therefore recapitulate the length of the cell-free DNA in the circulation. Larger fragments might indicate that cell-free DNA originating from blood cells was released during the pre-analytical process, during the plasma preparation, or due to shipping or due to storage. In terms of the cell-free RNA, they assessed the hemolysis by the amplification of the MER451 base pairs, MER451A expression, and the heme absorption, as we did. And they found that Norgen's tube and packaging tubes were the best regarding the hemolysis, showing no induction of hemolysis for up to 7 days. Ours didn't show induction in our hand for 30 days. And they assessed this, as I mentioned, by the MIR 451A. Roche tubes also performed well for microRNAs with an only slight induction of hemolysis, which hugely increased with STREC after 5 days. In terms of yields for the cell-free RNA, they found that the total cell-free microRNA content remained stable in blood stores for up to 1 week in Norgen tubes. STREC and Roche tubes showed contamination after 24 hours, and in vaccines, possibly due to the changes in the expression during storage. On average, Roche had the least yield, as they measured it by PCR amplification, with the 45 nanogram microRNA per ml of plasma. STREC and Norgen were comparable. STREC showed 79 nanogram per ml, whereas Norgen showed 86 nanogram per ml. And they concluded the following. Norgen BCTs are advantageous as they do not use formaldehyde fixation, which is a harsh treatment, and that is one of our main statements. Norgen BCTs are compatible for the extraction of both cell-free DNA and cell-free RNA, whereas other tubes have separate tubes for RNA analysis, making it costlier, not only financially, but also in terms of sample amount available. So you don't need to collect two blood samples, one for DNA, one for RNA. You only have one tube that can give you both DNA and RNA. Therefore, for this, Norgen tubes are desirable for any marker studies or combinatorial marker studies. Now, since we talked about the cell-free preservative, so I believe now we need to know how you should be isolating your DNA and RNA from your preserved plasma. Currently there are two purification technologies on the market: the phenylchloroform based technology and the silica-based technology. Phenylchloroform is an inexpensive technology, as we all know. It's mainly used for the purification of total RNA, but the drawbacks around it, the obvious one, is that if you have carryover contaminations of the phenol in your purified RNA, this will affect or inhibit your downstream applications. If you want to go to next-gen sequencing or microarrays, even for PCRs, regular PCRs, it will affect these downstream applications. Then there is the silica-based column, which is a clean system, but it's not very optimal for RNA. It's more for DNA. And silica-based columns were originally used for DNA purification, but it has some drawbacks as well. It has a molecular weight cutoff, so it binds certain sizes, and it has a bias towards binding large molecular weight DNA. When we combine both phenylchloroform to silica-based column, we're going to talk about the drawbacks right now, especially for RNA. However, Norgen doesn't use silica for the purification of DNA and RNA. In fact, we are using our patented silicon carbide, which is available either pre-packed in spin columns, as you can see in this small picture, or as a slurry format. And that slurry format is just to accommodate large sample volumes. We're gonna talk about the slurry format and the column format for the cell-free DNA isolation later. This silicon carbide It has lots of advantages over silica or phenochloroform. Silicon carbide doesn't have any molecular weight cutoff, so it covers or it binds all size ranges with no bias towards large sizes or small sizes. Also, it binds all sequences without a bias towards certain or specific sequences. Also, this picture shows for RNA that with Norgen tubes, we are actually purifying truly total RNA, the large RNA, the 28S and the 18S, all the messenger RNA with all of the sizes, and we're not missing on the small RNA, whereas silica-based columns, it's missing a lot of the small RNA. This is because silica, as I mentioned earlier, has a bias towards binding large sequences more than small sequences. I would like also to mention that in all Norgen purification methods, we're not using any phenochloroform because first of its drawbacks. Second, phenochloroform for RNA specifically, coupled with silica-based columns, it's going to have a bias towards binding sequences, especially RNA sequences, with high GC content. So it's going to be missing lots of RNA sequences, microRNA, small RNA, with low GC percentages. And that was mentioned in a letter to the editor in the molecular cell, showing this, that there were some microRNA were mentioned to be differentially expressed, like downregulated, and they were mentioned to be as a biomarker, and then it turned that actually they couldn't find it. That's why it was not bound, and they thought that it's now downregulated. So this is a major drawback for coupling silica with phenochloroform. In order to assess this ourselves, we did a small spiking experiment, but in depth, more than what we found or what we read in the paper. We spiked plasma or serum with sequences, synthetic sequences of RNA with different percentage of GC contents or GC percentages. And you can see that Norgen captured 30 unique microRNA with very low GC percentages, whereas the other competitors or silica-based columns didn't recover any of the small or low percentage of GC content. And this in full agreement with the publication that mentioned this. So now silica or silicon carbide, our technology, Norgen's technology, doesn't have bias towards binding any sequences, doesn't have bias towards binding any sizes. Therefore, this is perfect for cell-free RNA and cell-free DNA, because cell-free RNA and cell-free DNA is a small size. Also, it has varying sequences, GC percentages, so we are not going to be missing anything on these from these samples. So now, back to the cell-free DNA and our preservative and how we isolate the cell-free DNA from the recovered plasma from our tubes or from any other tube. So currently, Norgen is offering two formats for the purification of cell-free DNA or cell-free RNA. But for cell-free DNA, two formats. One is a slurry format and one is a column format. Both have the same outcome. The only difference that our silicon carbide is pre-packed in spin columns, so it's a column format. The slurry format, our resin or silicon carbide, is loose in a buffer. That's why it's called slurry. And the main reason is we have the slurry format because sometimes you need to isolate from large sample volumes from 10 ml or higher. Then with slurry format, you can go very high volumes. With the column format, you are limited by the size of the column. Also, the column format does have some extra steps to further concentrate your DNA into a very low elution volume. So if you started with 10 ml, you can elute in 25 microliter. So your DNA now is going to be super concentrated, and you can easily go to any of your downstream application. Whereas for the slurry format, you can elute in 50 or 100, and then we recommend another kit to further concentrate the DNA into a smaller volume. However, the quality and the quantity from both formats are considered the same. Also, the cell-free DNA purification is now automated on the Hamilton Nimbus Microlab platform. It's going to be soon on our web page, and you can purify your DNA along with the concentration steps on this machine, 24 samples at a time, and you can purify your DNA from as low as 200 microliter up to 5 ml, and there are options for going even higher volumes. So as I mentioned before, if we need more DNA, we automatically think of using more volume. So, sorry, this is a representation of the amplification of the 5S ribosomal gene from different sample volumes compared to a silica-based column. And we expect that whenever we increase the volume, the amount of the DNA will increase. So as you can see, with all volumes, starting from 50 microliter up to even 10 ml, there is a very good linearity, 90% or higher, which means whenever you go higher volume, you get the higher the concentration or the higher the amount of DNA. With the silica-based technique, at some instances it's linear, sometimes you lose linearity. This could be the loss of the linearity, maybe because of overloading the sample, the silica with DNA, or because maybe the quality is not as good. That's why you are getting inhibition. That's why you are losing the linearity. So if you went 50 microliter, the inhibitor may be less, but when you go higher volumes, you are increasing the amount of inhibitors. So you're gonna get DNA, but it's gonna be inhibited. You can go to your downstream application, obviously. Okay, this is a very important slide because most of us, when we isolate the cell-free DNA or the cell-free RNA, and before going to our downstream application, being PCR-based, being sequencing, microarray, SNP analysis, methylation studies, all of these things, we measure the concentration. And commonly used measurement tools, they have certain limits. And these limits are in front of you, like regular specs. It can go down to 10 nanogram. A nano spec, like nano drop or whatever, it goes down 1 to 10 nanogram. This is the limit of detection. Ribogreen or picogreen, it goes down to 1 nanogram. Agilent bioanalyzer, it goes to 50 picogram, depending on the chip that you are using. In plasma, serum or cell-free bodily fluids or liquid biopsy that doesn't have cells, the concentration significantly varies from sample to sample based on diet, sex, time of sample collection, exercise, health status, and the range varies between 1 picogram per microliter up to 100 picogram per microliter. So you can get from a sample 1 picogram per microliter, so or 5 picogram or 10 picogram. If you try to use like a nanospec or even Agilent bioanalyzers to measure the concentration of DNA that you have been purifying from 200 microliter of plasma, you're going to be getting maybe 10 picogram. And then when you go to the Agilent bioanalyzer, which has a limit of 50 picogram, you will see nothing. So you will think, I didn't isolate DNA. But actually there is DNA, but you can't quantify it because the quantification method is not optimal for measuring the concentration that is very low. Therefore, for such a reason, Norgen developed 2 kits to accurately quantify DNA and RNA, the cell-free DNA and cell-free RNA, but using a PCR-based method. And since PCR is very accurate and sensitive, this PCR methods for or procedures to quantify it can go down to actually 50 femtogram. So now, when you isolate your DNA or RNA from plasma from 200 microliter, you can confidently know the concentration using PCR-based. If you try to go to a nanospec or regular specs, all what you're going to be getting is a background. And from our experience, sometimes from the same, like if we are isolating from 10 samples, all samples will show us the same number, 40 nanogram, 50 nanogram, and we will say, okay, we have very good DNA. When you go to the amplification or your downstream application, you adjust your concentration or your PCR input based on the concentration that you are getting, and then you're going to be getting very high CT values or no CT values, and then you will think that you are not amplifying, you are not getting any DNA, or the DNA is inhibited, but actually you based your calculation or you did your calculation based on inaccurate measurement. So now, if you can't do Agilent bioanalyzer, or you can't measure the your DNA with nanospec for the quantification, then how are you going to check on the quality? Mainly, we check the quality by the 260-80 ratios. We check the quality according to the RAM value. And we know if we have good DNA or bad DNA, or good RNA or bad RNA for our downstream application. You are not going to be getting such quality or values from a PCR amplification. So in order to check the quality, we found that if you isolated your DNA from plasma or RNA from plasma, and you measured, as I mentioned, the concentration on a nanodrop, you will get very poor 260-80, which is mainly like around 1 to maybe, at best case, it's going to be 5. And you will say this DNA is not of a good quality. Actually, these numbers now, 260-80 doesn't mean anything, because in this application note, it can be found on our webpage, we did a small experiment, but it's very important. The results that came from this experiment was very important. We found that whenever you have low concentration, the 260-80 will decrease based on the concentration. So if you started with a very highly pure DNA or very highly pure RNA with very high concentration, very high or very good 260-80 or very good RAM value, and then you serially diluted this RNA and measure these serially diluted RNA samples, you will find that whenever you go lower in the concentration, the 260-80 will decrease. When you go to your lowest concentration, the 260-80 will be very bad. But this RNA has initially came from a very good RNA quality, which means that whenever you go down in the concentration, the 260, 280 will be low. But if you took this RNA at the lowest concentration and you go to your downstream application, it's gonna be perfect for any downstream application actually. So now, if you are deciding on your quality based on 260, 280, especially if you're isolating DNA from plasma or serum or RNA from plasma or serum, this will not give you a good indication about the quality. In order to know actually the quality of your DNA or your RNA, we recommend being also PCR based. If you took your purified DNA or purified RNA and you did amplify any target of your interest, but use increasing amount of PCR input, like from the same purified DNA, use 2 microliter PCR input, 4 microliter PCR input, and 8 microliter PCR input. The more PCR input or the more volume you add from your DNA, you should be getting lower CT value. If it declines, like in the graph in front of you, like this, which means that your DNA is pure, and with a good quality. If the CT value started high and then dropped and then become flat, then you have a little bit of inhibition or inhibitors. If it stayed flat all the time, then you have DNA with very high amount of inhibition. If you didn't get any amplification at all, which means that either you don't have any DNA or you have a lot of inhibitors in your sample. This is the most accurate and reliable way to assess the quality of your purified DNA or RNA. And it's very accurate as well. So now, I would like to discuss or show a very good publication that has been published like 3 years ago, March 2015. It compared 2 of Norgen's kits. One was a slurry format and another one was a column format against 9 different methods for the purification of DNA from plasma. And also some published procedures. What they found that Norgen's kits on the panel number A, or like the upper graph A, it recovered the highest amount of DNA from different plasma samples collected from different cancer patients as well as healthy individuals. Whereas Qiagen recovered less DNA from all of the samples. If you notice the scale, ours goes to 140, it goes to 180. If the scale at this graph is 140, you're gonna see like very big difference. And the best thing they did in this paper, that they based their quantification, or their quantification was based on 3 different assays. 2 of them was a qPCR assay and then a qubit assay. And the 3 different quantification methods showed the same outcome. Norgen's kits with the silicon carbide recovered the highest amount of DNA in compared to all the other 9 procedures, being commercially available procedures or published procedures. Another key comparative paper, they they compared two spin columns kits or two spin columns format, Norgens and QIAM circulating nucleic acid versus 4 different magnetic bead-based formats. And the best thing that they did in this paper is they spiked their plasma with a ladder, DNA ladder, with different sizes ranging from 25 base pairs up to 800 base pairs. And they wanted to actually check the efficiency of the different isolation methods on recovering the different sizes. Since cell-free DNA in plasma is small size DNA, 140 to 180 base pair, so they want to know which one of these methods will recover this, the size of interest, or what is mainly going to be in the cell-free DNA. Collectively, the recovery of all sizes, the spin column format being Norgen or QIAGEN was insignificant, no significant difference, but spin column format being Norgen or QIAGEN were significantly better than any other magnetic bead-based isolation. But when you compare these two, like that was not clearly explained in the publication, but I'm going to show it to you. Okay, so this graph shows the efficiency of the recovery of all of the sizes of the DNA. From the first glance, you can see that the recovery of all sizes using magnetic beads is maximum around like 60% recovery. And this is a spiked amount, which is high. So it's whatever amount it was spiked is going to be actually higher than what is normally found in cell-free DNA, since they lost, or magnetic bead-based kit lost 60% of a spiked high amount material. So this will be amplified, or the percentage will be higher. The percentage of loss will be very high if you are isolating cell-free DNA, normal cell-free DNA from plasma. But when you compare the two spin columns, Norgen and QIAGEN, you will see that Norgen recovered the sizes of interest, actually from 50 to 100, even 150, recovered it with a very good efficiency, whereas QIAGEN, which is based on silica columns, and as I mentioned before, it has bias towards binding large genomic DNA sizes, or large DNA sizes, which is mainly in plasma samples. That high genomic DNA is considered contamination, genomic DNA contamination from hemolysis of the red blood cells and white blood cells. And they have silica columns has very poor recovery of the small DNA sizes, which is the DNA size of interest for any researcher who is interested in doing his work on circulating tumor DNA and circulating free DNA. So there was a systemic drop in the recovery below 150 base pairs with silica column, but with Norgen silicon carbide, it showed better recovery of DNA fragments of 150 base pairs and below. And this also will bring us back to the RNA, which is better diversity. We don't have any bias towards any sequences. We don't have any bias towards any sizes. So out of these 3 comparative publications, one for the cell-free DNA preservation and cell-free preservative, Norgen cell-free preservative, and the 2 publications evaluating Norgen's silicon carbide technology for the purification of cell-free DNA from plasma compared to silica-based technology being spin column or magnetic beads, we can conclude from these 3 publications that Norgen's tubes, as I mentioned before, are compatible, or as mentioned in the publication, compatible for the extraction of both cell-free DNA and cell-free RNA, whereas Other manufacturers supply separate tubes for RNA analysis, making it costlier, not only financially, but also in terms of the sample amount. Norgen tubes show the highest cell-free DNA yield. Norgen tubes are the most desirable tube for biomarker research. And Norgen purification technology, which is based on silicon carbide, showed the highest DNA yield as compared to other silica-based columns, being spin or magnetic beads-based. And Norgen cell-free DNA purification technology doesn't have any bias towards certain sizes as compared to silica-based technology that favors binding large DNA sizes. So the outcome out of the 3 comparative studies that coupling Norgen cell-free preservative with Norgen's technology which is based on silicon carbide for plasma cell-free DNA is the new gold standard for cell-free DNA preservation and purification. This is based on recent publications. So thank you so much for joining our webinar. And if you want to contact us, you can reach us through our webpage or the emails in front of you or by phone. And also there is a promo. You can receive 50% of your first order if you use this promo code. Thank you so much. Now I can receive any questions. Okay, no, that's fine. So I have 4 questions so far, increasing. So will you send the slides after the presentation? I believe we can do this. And the second question, in your Agilent trace, the cell-free DNA from your tube has a length of about 160 instead of 146. How do you explain this? Actually, it depends on the sample that we are isolating. Sometimes we are isolating from different donors, and different donors will have different sizes depend on their sample type. Like it's variation. It ranges the range from 140 to 180 base pairs. The lowest in the 140 to 150 or 160, that's the best thing. If it's 180, that's fine. But if it's more than 180 or it's duplicates showing an apoptotic ladder, this means that we are now getting degradation. But between 140 and 180, that's fine. Okay, and then can you please go back to the previous slide for the advantages? Okay, Ambient versus Norgen versus Qiagen. You see content slide, what Qiagen? What Qiagen's kit was used? It was the microRNA Easy kit. The cost of the tubes and column-based cell-free DNA purification kit, you can reach, as I mentioned, one of our marketing and sales team by email or phone, and they will be more than happy to discuss with you the cost. Unfortunately, I don't have information for the cost. I'm just the science guy. In amplifying cell-free DNA, what is the minimum concentration for DNA amplification? Only 10 nanogram works in our lab. Okay, this is a very good question. Okay, as I mentioned, for the quantification. I don't know, how did you quantify your DNA? Because you mentioned that you did 10 nanogram. I believe it was measured based on either nanodrop or qubit. And for both methods, as I mentioned and as I showed on the slide for the quantification, the limit of detection is of these methods are very low, and the DNA actually in the plasma is lower than the detection limit of this measurement tool. So this 10 nanogram, I don't know if it's very reliable or not, but in our hand, we don't measure the concentration because it's very low, and we check the quality using, as I mentioned, the increasing amount, and the quantity. We use the low abundant DNA quantification kit that quantify the DNA using PCRs, and normally the concentration is going to be around 500 picogram, maximum 1 nanogram. When we do amplifications, even without to test linearity or to test these things, we use one volume, like 2 or 3 microliter in a 20 microliter PCR reactions. And since we are doing this every day, we know what CT values are we going to be expecting. It varies, of course, because the cell-free DNA amount will vary from sample to sample, but we have like a range that we are expecting our CT values will be. You can reach us via email. You can reach me directly on my email, and then we can discuss this in details. I want to know what volume did you isolate your DNA from? How did you amplify? How did you quantify? What target are you amplifying? Because all of these things matters. 10 nanogram actually, when you say 10 nanogram works in our lab, actually you can, when we do our services here, because we have a service facility for sequencing, and we isolate RNA from as low as 50 microliter of plasma, where the amount of RNA is very, very low. We don't quantify, we go directly to our sequencing after doing some quality control checks before, but the concentration sometimes is in the femtogram range, and the PCRs works. So if you are having like concentration of 40 nanogram and you're trying to go down to 10 nanogram using based on this quantification method, then it's not going to be reliable calculations. Actually, you can amplify DNA from like in the femtogram range. So I don't know why exactly only 10 nanogram works in your lab. So there is another question, but this is going to be answered by our marketing and sales team about the time to receive in Mexico and order of tubes. They will take care of this 100%. Okay, Asian traces are donor dependent. Okay, but in German publication, they say the mean is significantly different. Yes, the mean is significantly different because everyone was 183 base pairs, only Norgen was 143. This was in their hands with their samples. Actually, again, it's donor dependent, but I think 143 base pairs or 146 base pairs agrees more with all literature than that saying that the cell-free DNA or the nucleosomes or the chromatosomes are within this range. Actually, circulating tumor DNA, not cell-free DNA from normal cells, but circulating tumor DNA has even lower sizes than the 140. I believe it's 135 base pairs. So the range varies from 140 to 180 according to the literature. The nucleosome or the chromatosome, which is the DNA wrapped around the histones, is 146 base pairs and its duplicates. They said yes, significant difference between the sizes, but which size is agreeing with the literature? Is it the 180 or the 146? I believe all the literature is saying the 146. Sometimes we, like I see samples that I isolate and check on the BioAnalyzer, it's 170 base pairs. Again, it's donor dependent. I don't say that we are significantly better. They are not significantly better. No, the same, but the literature is inclining towards our sizes, according to the German publication. For SNP mutation, actually we don't have currently data for SNP mutation, but we are working on it. So once it's ready, we're gonna publish it. And since our preservative is fairly new, it has been on the market for I believe a year and a half or two, then this publication that was published 2 weeks ago was the first publication featuring our BCT tubes. I believe there are going to be more coming because lots of people have been trying this preservative tubes, and I believe they are in the process of showing their data. The low abundance quantification kit are for human nucleic acid. Yes, it's for human DNA because the targets, because in the low abundance kit, quantification kits being RNA and DNA, we amplify specific targets that are mainly human targets, human sequences. And the recovery of increased plasma volume in the cell-free DNA of Norgen is due to less hemolysis, right? Yes, the recovery of increased plasma volume is due to less hemolysis, less movement of the cells. The chemistry behind this preservative, once the blood is mixed with the plasma, the chemistry pulls down the cells instantly without even centrifugation by gravity and prevent it from moving. That's why the volume is constant. We don't lose any plasma volume. We don't lose any plasma. Actually, yes, we are currently analyzing some sequencing results for RNA, and we're going to be publishing it soon. Okay, yes, we did actually isolated cell-free DNA and microRNA from the same sample, and you can go as low as 50 microliter. We have kits that can isolate down to 50 microliter of DNA and RNA up to 10 ml, and the amounts of the RNA and DNA purified from these low volumes are amplifiable and good for any sequencing. Also for sequencing, we do have a very good platform and workflow for library preparation from challenging samples like plasma. That can we do lots of services from low sample volumes for RNA. We do our library prep using our kits, and we are using which is compatible with the Illumina platform. Okay, so there is a question: how can you get 6 to 7 ml from only 8.3 ml of blood, whole blood? Is it that unusual? Actually, it's not very like, it's based on the chemistry, and now we have like the preservative in the tubes is 1.5 ml. You have 8.3 approximately 8.3 to 8.5, because the cells, the chemistry is compacting or the preservative is compacting the cells at the bottom of the tube, so you are recovering everything, like most of the liquid of the blood, because the cells are very compacted at the bottom. When you collect your blood on EDTA tubes and you spin it down, half of the tube would be the blood with the buffy coat in the middle, and then the plasma volume, which is going to be 3.5 to 4. If you try to move the blood again in the EDTA tubes or any other tubes on the market, by the way, and the blood pellet will move and will go back into solution. But in our case, once you spin it down, the blood pellet is going to be solid at the bottom, and if you try to move it back again, it's not going to move. So because it's very compacted at the bottom. Oh, this is for the RNA kit. Okay, so this question is, could you clarify the difference between the plasma serum RNA mini kit column format versus the plasma serum RNA slurry format? The plasma serum RNA mini kit with a catalog number of 55000, it's a spin column format. It goes up to 200 microliter of plasma, and it can elute your RNA as low as in 10 microliter and up to 25 microliter. So it has a volume range up to 200 microliter, and it's a column format. The plasma serum RNA slurry format with the catalog number 42800, it's a slurry format that can go as low as half a ml of plasma, not lower than half a ml of plasma, and up to 5 ml of plasma. It's a slurry format. The elution volume is a little bit high, 50 microliter up to 100 microliter. And then if you would like to concentrate this 100 microliter or 50 microliter, we recommend also using, for the concentration step, Norgen silicon carbide technology. We have an RNA cleaner and concentration micro elute kit, where you can take this 100 microliter, concentrate and elute in as low as 8 microliter. And then if you want to do sequencing, you can take these 8 microliter, go right away to your library prep and do your sequencing. Yes. So the new Norgen tube, can you still isolate the white blood cells buffy coat? Okay, this is a very good question. So in our case, with our tubes, because the chemistry is unique, completely different from other tubes, when you spin down the plasma, when you spin down the blood and recover the plasma, you will not find a buffy coat. The red blood cells and white blood cells are going to be in the pellet. We have a supplementary protocol on our webpage showing how you can isolate the white blood cells, or that if you want it intact, you can isolate it. If you want to get the white blood cells following by isolation of DNA from these white blood cells, you can do it. We have a supplementary protocol that is published on our webpage right now for this application. Okay, so this question also similar to the previous question. So how you can get out the CTCs from this compact cell pellet? Okay, at the beginning, and this is also on our webpage as a supplementary protocol, from the cell pellet, we have a buffer called RBC lysis buffer, where you lyse all the red blood cells, and you're gonna be left with the white blood cells and the CTCs as a very tiny white blood cell, as a very tiny pellet, white pellet, and then you suspend this pellet in your buffer of your interest, and then you can do your test if you're going to do flow cytometer, or if you want to do isolate DNA from these CTCs or white blood cells. We have a supplementary protocol similar to the previous question. We have a supplementary protocol for this application on our webpage. Okay, is there a slurry format for circulating tumor DNA? Okay, so our purification method purifies DNA in plasma, circulating DNA in plasma. The circulating DNA covers circulating free DNA coming from apoptosis or circulating tumor DNA coming from necrotic tumor cells. So it's total DNA from plasma. And this total DNA means any DNA in plasma will be recovered using the slurry format or the column format. So there is no specific kit for cell-free DNA from normal cells, and there's going to be a specific kit for circulating tumor DNA. You're going to have both in one pool, all the normal DNA and or plus the circulating tumor DNA. Could you share with us your email address in case more questions? Yes, of course. My email address is Sando. Okay, so my email address is Momen, M-O-E-M-E-N, @norgenbiotech.com. Okay. I believe we are done here. No more questions, and thank you so much for joining our webinar. And if you have any questions or you need further clarification, please email me at my email address, moemen @norgenbiotech.com, or you can email us at info@norgenbiotech.com. Thank you so much.